I did my project on the resistance of Salmonella typhi and Salmonella typhimurium. Many people have heard of Salmonella, but many do not know that there are different types of Salmonella bacteria, and some strains have become increasingly resistant. The bacteria Salmonella typhi is rod-shaped and is aerobic, which means it needs oxygen to live. It stains pink because it is gram-negative and has 3-layer cell membrane. They also have flagella that make them motile and help them swim through the intestinal tract. The illness that this bacteria causes is typhoid fever, which can come from contaminated food or water. Treatment is necessary for this illness, and without it, liver damage, heart damage, and internal bleeding can occur. It only infects humans and is not common in the United States, but it is common in underdeveloped countries and low-income countries. Salmonella typhimurium is also rod-shaped and aerobic and has flagella. The flagella swim through the intestinal tract and fight with epithelial cells to stick to the intestinal surfaces. This bacteria causes Salmonella diarrhea, which can lead to vomiting, fever, and stomach pain. Treatment with antibiotics is not always useful or used for every patient, but when a patient has a compromised immune system or is resistant strain, it is necessary. This bacteria can be transferred through the ingestion of raw or undercooked meats and eggs. Both of these bacteria present many different genes, and scientists can use DNA sequencing to understand the strains that cause a particular disease and how the disease should be treated. Using DNA sequencing lets the scientists know if the bacteria has changed and how the bacteria adapts and lives inside their host. The use of common and limited antibiotics has caused some Salmonella strains to become resistant to the antibiotics. The research on resistant strains have proved that these bacteria are learning to survive in their host and now contain antibiotic-resistant genes and also have lost gene function. An antibiotic resistance pattern study on S. typhi was performed in Bangladesh to test the resistance and sensitivity of the bacteria to the antibiotics used to treat the condition. The isolates in this experiment was tested using 12 regularly used antibiotics. The results of this study showed that the highest resistance was against nalidixic acid, and resistance depends on the age of a patient. The rate of resistance was the highest in adults 30 to 40 years, but there were also samples from children that had resistant strains that were resistant to all antibiotics. Factors that contribute to antibiotic resistance may be due to drug overuse, misuse, and inappropriate practices by physicians. Another factor due to resistant strains can be antibodies in animals that transfer to humans when humans consume their meat. Lower income countries are more prone to antibiotic resistant strains due to the lack of doctors and the right antibiotics, and they are not aware of resistant strains. This research concludes that ciprofloxacin and nalidixic acid are not a treatment option anymore due to resistant strains, and the patterns of resistant strains due to geographical and environmental variants. Characterization of antimicrobial resistance markers and their stability in Salmonella typhi research was conducted to characterize resistance plasmids based on their transferability and stability. Multi-drug resistant S. typhi strains contain a self-transmissible INC-HI1 plasmids that carry genes causing resistance to first-line antimicrobials. Plasmids are genetic structures in a cell that can replicate independently of the chromosomes, typically have a small circular DNA, and are used to manipulate genes. The INC-HI1 plasmids are known to be quite large and thermosensitive. Our plasmids found in this study are not part of this group and are small and have no thermal sensitivity. The results showed that all of the resistance markers were transferred through conjugation and transformation. None of the resistance markers for nalidixic acid were transferred, which is probably due to its chromosomal origin. Multi-drug resistance in S. typhi associates with high molecular weight and a self-transferable plasmid that transfers the phenotype. Resistance plasmids found on S. typhi are small and highly stable, causes a great concern because of its transmission potential and the increased spread of antimicrobial resistance. Clinical settings must watch for the increase of antimicrobial resistance to prevent further spread of transmission. In this study, Salmonella typhimurium strains were isolated from the retail ground meats found in the Washington, D.C. area. 84% of the isolates were resistant to at least one antibiotic, and the S. typhimurium strains found were resistant to 5 antibiotics. These strains were found in pork and chicken. While giving antibiotics to domestic livestock that are meant to prevent and treat diseases and promote growth has caused the emergence of antibiotic-resistant bacteria. The antimicrobial agents are transferred to humans through the food chain. Antibiotic-resistant integrons were found on all of the S. typhimurium strains. Integrons are genetic mechanisms that allow bacteria to adapt and evolve rapidly through the differential expression of new genes. Depending on the size of the integrons and the particular strain determine what antibiotic they were resistant to. As the number of cases increase, it is caused a concern considering the extreme resistant phenotypes of S. typhimurium and the fact that it is found in retail meats. More efforts are needed to reduce the increased spread of this resistant salmonella. This study gave a closer look on the MAC-AB efflux pump in Salmonella typhimurium. There are many multi-drug efflux pumps, but this is the only ABC type. Resistance to antibiotics can be due to mechanisms such as drug modification, target modification, and inhibition of uptake. The first line of defense of this efflux pump is to efflux antimicrobial compounds. The MAC-AB efflux pump is required for survival in the inflamed intestine and macrophages because this is where the bacteria is exposed to highly toxic reactive species. In macrophages, Salmonella typhimurium creates a vacuole that interferes with vesicular tracking of NADPH. Without this efflux pump, severe attenuation of the bacteria occurs. The results showed that the MAC-AB drug efflux pump is involved in the protection of Salmonella during oxidative stress and is required to infect hosts. The research on this efflux pump is useful in understanding what the bacteria can and cannot resist and how it prevents antibiotics from killing it, and can be useful to scientists to create helpful antibiotics for the future. In conclusion, physicians need to be more cautious when giving antibiotics to patients. The misuse of antibiotics has caused S. typhi resistance in a threatening way. Antibiotic resistance is now common among children due to this misuse. Resistant plasmids cause a huge concern because they can exist in their host and easily transmit and spread resistant S. typhi. Clinical settings need to keep up with the antibiotic resistant trends to stop transmission and spread of the bacteria. The research has proven that the food supply is a major source of antimicrobial resistant S. typhimurium, which is why new guidelines for retail meats are needed. Retail ground meats contain reservoirs of resistance from animals that are then transferred to humans, which is why a surveillance program on foodborne pathogens would help prevent the spread of the disease. More research on the MAC-AB pump would be beneficial for scientists to create better antibiotics. S. typhi and S. typhimurium have become unique due to their evolving strains and resistant strains.